Everyone, I'm Miles Skoda, and this is the OutsourcePharma.com webinar series. And today we're demystifying large-scale continuous chromatography for small molecules. Now, joining me for this presentation is an industry expert from SK Pharmteco, head of R&D engineering, Dr. Olivier Dapremont, who is going to provide a basic understanding of the simulated moving bed technology and look at its benefits compared to other technologies, including rapidity of development, easy scale-up, CGMP compliance, cost-effectiveness, and superior sustainability through recycling strategies. Now, we will be holding a Q&A session following this presentation, so please feel free to submit questions as we go along. We'll get to as many as possible during that Q&A, but any that we don't get to, I will pass along to the team here for potential follow-up after the event. And then just a bit of housekeeping: if you run into any issues today, please try refreshing your browser first. If your problem persists, you can submit a question through that Q&A, and I'll help get you back on track. And then finally, this webinar will be available on demand, usually within a day, often much less. You'll get an email with a link to it, or you can just use the same link that you used today. So before we get started, we just have one quick poll question. All we're asking is, have you used chromatography as a production technique? Feel free to answer, provide as much context or detail as you want. And with that said, I'm going to turn it over to Dr. Dapremont to get started and bring us in with the agenda.
Well, thank you very much, Miles, for the nice introduction. Welcome, everyone. Good morning, good afternoon from wherever you are located watching this webinar. Like Miles said, we're gonna address some issues with or some perspective on simulated moving bed or chromatography for large-scale manufacturing. So the agenda for today is first a quick introduction on SK Pharmteco, the company, or the corporation. Then we'll talk about prep chromatography as a technology, and then we transition to simulated moving bed or continuous chromatography principle to understand how this works, a little bit more on the mechanics of it. And then once we understand the process, then we can look at how we develop separations, specifically for chiral separations, how we can go very rapidly to a process that can be scaled up. And then finally, we'll talk about some applications and some consideration, how to implement the technology at larger scale, and the benefits of it. So SK Pharmteco, like I said, a couple of slides on the corporation. So we're part of the SK family. SK is one of the largest corporation in South Korea. The SK Pharmteco is the pharmaceutical branch with three divisions: small molecule here in the US, in California and Texas, and then Ireland and Korea. We also have a division that does cell and gene therapy based in Europe, in France, and then in the US on the East Coast. And then an analytical development branch, which is based throughout the all different facilities. So we combined about over 80 years of experience in large-scale manufacturing of APIs under CGMP, obviously, and with about 2,000 team members across the globe and 6 manufacturing sites. So what do we offer? Like I said, we do the process development, scale up to manufacturing APIs, clinical and commercial manufacturing, obviously. We do regulatory support because we manufacture in the CGMP. And we do through differentiators, technologies like energetic chemistry. There's a long history of energetic chemistry, specifically here on site in California, formerly Aerojet, well known for handling very hazardous chemicals. We obviously, spanning out of that, we have a lot of continuous flow processing capabilities here in California, but also in our site in Korea. We do handle high potency throughout our different facilities. We do catalyst design in our South Korean facility for high pressure trickle beds type of hydrogenation and other type of catalytic reactions. We do have particle engineering throughout our sites, but with a center of excellence in Ireland. And then obviously chromatography, which is today's subject, with the center of excellence here based in California with both batch and simulated moving bed capabilities, SMB being one of the largest capabilities installed since the early 2000s here. Like I said, we are located at 6 different locations throughout the whole world. We have sites in California and Texas for small molecule, also in Sejong in Korea, and then in Ireland, outside Dublin, at the Seoul site for the small molecule business. On the large cell gene therapy, we have 2 sites, one outside Paris, at Corbeil-Essonnes, and the one in King of Prussia, Pennsylvania, the former GSK site. We have a commitment to ESG, and through technology, we try to achieve the best performance in terms of waste minimization, recycling, and carbon emissions. So we believe that through technology we can achieve our target of net carbon zero by 2040. And SMB is one of those technologies that provide very good capabilities for solvent recycling, solvent reuse, minimizing waste, and reducing your impact to the planet. So that was the a quick introduction on the company. If you want to learn more, please feel free to reach out, log on to our website, our new website, or reach out to our BD people. So today, what we're trying to address with this webinar is the stigma associated with chromatography. Chromatography is always perceived a very, very expensive technology because usually it's very misunderstood by a lot of chemists, but we can achieve very high yield, very high purity, and very high throughput with equipment, with automation, and continuous technologies that are available for several years now. So that provides very competitive pricing for projects. There's limited applications. Well, SMB or chromatography is used for many purification process, and a lot of things have been done using SMB. Development is not as difficult as most people think. Most of the chiral separations are being brought up to commercial. I've been developed in less than 2 weeks with a very fast scale up. So scalability is also one of the benefit of the technology of chromatography. So all those things are actually very beneficial for the process. And obviously we can do a very large commercial scale on GMP, and with recycling strategies, we have sustainability aspect that's not negligible these days. So hopefully at the end of this webinar, I would have convinced you that continuous chromatography is a technique that you should consider for commercially processing. It's cost effective, it's fast development, so get your product faster to the market and give you a competitive edge compared to your competition. Keep pressing the wrong button. So obviously to go into the webinar, we have to assume that the attendees or the participants have an understanding of chromatography, the basic principle of chromatography. I expect that you have a little bit of knowledge of high pressure chromatography. I'm not sure if you're familiar with dynamic axial compression columns, but really understanding chromatography large scale will help a lot for this presentation. I assume you're familiar with efficiency, the number of plate counts, what does that mean? How column is efficiency, how do we measure this? What is the retention time, retention factor? Those are parameters we use for scale up. What is the difference between selectivity and resolution? How we can affect those parameters by playing with the flow rates and so forth. And then obviously for SMBs, it's crucial to understand the effect of the flow rate on the efficiency, but also on the retention time, because we play with the flow rates to be able to direct the product in the streams of interest. Now, chromatography is a very powerful purification technique. It's a technique that allows us to separate pretty much everything that is otherwise very difficult to separate. It's a liquid-solid equilibrium-based process. So we have a solid base, and then we pass product over that solid, and we rely on the difference between the retention or the affinity between the molecules and the solids to provide the separation. So obviously this is used in analytical. We can separate things that are very closely related, and the goal is to be able to separate things to be able to measure purity. But also that can be used for prep or production, preparative chromatography, where now the goal is not to separate everything, it's just to separate the product from the impurities. So we can bundle all the impurities on one side or the other of the peak and collect the product at the target purity with the highest amount possible in the slowest or the smallest amount of time possible, which is basically maximizing the production rate of the process. So every chemist has an HPLC or has used an HPLC in the lab. It's ubiquitous, it's everywhere. Analytical tool by excellence. It's combined with other technique. It's used to identify impurities, measure purity of product. So we do use chromatography in liquid or in gas or in supercritical all the time in analytical world. Now, it's also used in production. Sugar purification is done a very, very large scale, built it on a day using ion exchange column in a process that's very similar to SMB. It's called ISMB, intermittent SMB. It's a modification of the standard SMB system, but it's used at very large tonnage. Now, obviously, APIs are purified by chromatography, and also insulin and biologics and large molecules and peptides and so forth are in need of chromatography, the different types that exist out there. So it's a very powerful tool to use. What does chromatography provide? It provides a really efficient solution. We can get really high purity. We can target greater than 99% purity, especially chiral separation. We typically achieve 98 99% chiral purity. Very high recovery. You can actually adjust the parameters of the process to tune your recovery. You can recycle fractions and so forth because it's a non-reactive system that allows you to maximize your product recovery. The trade-off here will be the throughput, how much material you can process per unit of time. And then one of the things from a chemical engineering standpoint, which is great, is that it's a linearity of the scale-up. Everything is based on the velocity at the particle size. So when you want to increase the quantity you process, you increase the diameter of the column and you increase the flow rate according to the cross-section to maintain that velocity constant. So maintaining everything else constant, the particle size, the bed length, etc., you will achieve the same separation, but with a bigger column. So when you do develop, like here on a 5 centimeter column, you multiply by 16x the flow rates to get the same separation on the 20 centimeter dark column. And that's pretty much it. And then development that way, it becomes extremely fast, and we can achieve very quick turnaround. Now, family of chromatography, we have batch and continuous. To take a batch process, make it continuous, we have to sacrifice something, what we sacrifice is the capability of collecting multiple fractions. We only get 2 streams coming out of the SMB, so it's a binary-like process. And obviously, all chiral separations are the main applications for this process. So enantiomer is a big application. By extension, diastereoisomers, if you can manage to get the diastereoisomer of interest first or last. But we have also done specific impurity removal. We've done natural product where you have a forest of peak and you're trying to remove a bunch of the undesired impurities. And then we have done some SMB mining, which is basically go fishing the product in what is otherwise a waste stream, a filtrate from a crystallization, for example. And obviously, in the large molecule field, there's a lot of interest in developing continuous processes because of the interest of lower cost, faster cycle time, etc., in associated with continuous process in general. And peptides and oligos and all those protein separations are looking to continuous. So now that we have put the basis of batch process, let's look at what simulated moving bed is and how we can achieve this kind of continuous process efficiently. So again, the same advantage, SMB is not much different than batch chromatography. We get high recovery, we get high purity, we can tune the parameters of the system to get the purity and the recovery we want. If you can racemize the undesired enantiomer for chiral separation, you basically make the product process zero waste process. And then because of the nature of SMB, it's an isocratic separation system. So you have one solvent composition, could be a binary or a single solvent. We'll talk about that in a minute. You can actually have strategies to recover and reuse the solvent within the process. So there is internally already an internal recycling of the solvent through the columns, but there's also you can actually take the product, concentrate it, and recycle the solvent and achieve close to 100% solvent recycling. And we have some numbers at the end of the presentation. So to transition to the SMB, we need to understand the batch first. So what is the batch? We have a column packed with a media, and then we inject a mixture of blue and red component, purple here, and then we push with an element, and the blue component is more retained than the red. So we start to see that the red is moving away from the blue. And as you see, the part of the column that's active is the part where the purple is present. So only a small part of the column is functioning over time as we doing the injection. And then you need to have a large column, long column, a lot of packing material to separate a little bit about material. Obviously, here we could do a second injection before this injection is out, which is called stacked injection to optimize the process. But you can see that there's an inherent inefficiency in the process. You inject and you wait to get the product out with a certain amount of material at the time. So how can we do that better? Well, we start with the same concept. We have a column, we inject a mixture of two components, and as we separate by pushing with the element, now I'm going to move the column in the opposite direction of the flow. So now, to take an example and forget about the units, but let's say the red component is moving at a velocity of 10 towards the right, the blue component is moving at a velocity of 2. If I move the column at a velocity of -5, minus being going to the left, now the red component is moving at 10 minus 5 at +5, so it's still going to be moving to the right with the element, while the blue component is now moving at 2 minus 5 at -3, which means for an observer that's located outside the system, looking at the feed position here, I can see that the blue component is actually moving backwards, moving towards the left, right? So when we do this, well, we create a gap at that point, at that location here. So because we have a gap, then we can fill it with more material. So we can, instead of doing an injection, now we can pump feed material into the system. So we have a continuous flow of feed material. And that's really a critical aspect, because now, just by doing that, we potentially increase tremendously the throughput. So unfortunately, there's a point where if you don't do anything, the column is going to be full, you're going to reach the end of the column, your system is going to just run out of space. So what you do is you create a bleeding of the material out of the system. So when you do that, you actually change the flow rate in the different sections. So here, and I'm going to use my pen here, red, in this zone here, the flow rate in here, it must be much higher than the flow rate in here, because we are pushing with more element to reverse the velocity of that blue component. We need to push hard enough that now the velocity of the blue component is faster than the negative velocity of the column. So then there's only an option, is to come out at this point. On the zone 2 and 3 here, we have a separation going on, so we need to make sure that the flow rates are tuned properly. So the blue component is still moving with the columns, not pushed hard enough by the element, and the red component is actually faster than the column, so it's going to the right. So we have separation. So we potentially have all that area where we can have all purple to as long as we get blue and red separate at the end. And then in zone 4 here, we need to take solvent out of here, and then when we take the solvent out, the flow rate out of here, the flow in this zone 4 is low, which means the red component is going to move at lower velocity, which has to be below the velocity of the column. So now the red component is being entrained by the column to the left. And then we keep adding little pieces of column over here, and then the piece of column that come out here, we can recycle it in the system. So we break the long column into smaller columns that are all identical, and then when this column is clean from the element pushing out here, when this column is clean, we can take it and move it in this position here. Now the solvent that's coming out here is clean, so that solvent also can be recycled in the process here. So now you can see we have two recycling loop, if you will. The packing material get recycled in the system. It's kind of a merry-go-round of columns going around, and then the solvent goes around the system. So we have, by this process, we have actually a much higher concentration of extract, and extract being the most retained compound, and the raffinate being the faster compound coming out of the system, and we have an inherent recycling in the system. So compared to batch, we have actually a better process. So another way to look at this is now you understand we have different zones. So we have 4 zones. Here in this case, we have 1 column per zone. The element is going from one column to the next, one column to the next here, and then this solvent here will go back here. Now, if I start, clear this, if I start the feed into the column in position 3 here, we can see the red components moving faster, eventually reach the bottom of the column. And if I have a detector here on the raffinate and an extract stream, I can see that on the raffinate stream, I'm getting a signal coming up, because now I have a red component coming out here. Now the flow splits between the raffinate stream and the next column, but remember, the flow rate in this column is much lower, so that front is not going to move as fast in the column as it was moving in here. So now if we don't do anything, the blue front that's somewhere here is going to reach the bottom of the column, there's going to be a problem because now I'm going to collect in the raffinator just a dilute feed. So what we do is we move the column by one position. So the column that was here has been moved there. So now that column is full of product to be separated. The column that was here has moved here. So now we have this column has already a little bit of red material in it, and is going to be fed with more material. Yeah, you can see the raffinate stream. Now it's only solvent coming out, so the signal goes back to baseline. Now, if you keep going in time, we can see now this column being pushed with a fresh element. We see the blue component separating from the red, moving faster. Eventually, the red component in column 3 reaches the bottom of the column, so we start to see a signal coming up here. We're getting more material in the raffinate stream, and we get to the situation where we have to switch again because the blue component is coming in. So now the column that was in this position has moved here, and now this column is being pushed out. The material in this column is being pushed out faster because we have a higher flow rate. So the material goes into the extract stream, and you can see the signal here popping up. Again, the signal in the raffinate is going down to baseline, so we have only this element coming out. And as we, and I'm going to keep moving a little bit, as we go down in time, you can see that eventually the blue component is clear of column 1, and the red component is starting to come into the raffinate stream, and we do a switch again, and we keep going, and we get a steady state. So when we reach a steady state, you can see that while we have continuous stream coming out of the system, extract and raffinate in terms of flow rate, in terms of concentration, we have actually a front of raffinate, a front of product, and a tail of extract, the tail of product coming into the stream in a position of phase here in frequency. This is a continuous flow of feed here. This is a continuous flow here, and this is a continuous flow here. So it's a continuous process, but it's a cyclic concentration result. And we can keep doing this until we reach the end of feed, and then we're done. So I'm going to skip this to get to the next slide. So as a result, this is what happens. We move the column by one position to the left here, but obviously we cannot move very big columns very efficiently. So what we do is we actually do, I'm going to start a video that you should see coming in your screen. We'll do what appears on the right here. You can see that the concentration profile is moving towards the right. And as the concentration profile moves to the right, we move the inlet and outlet port, extract, raffinate, feed, and eluent, by a column every time it's necessary. So when the red component is, or actually the blue component is reaching the raffinate port, we move the raffinate port to the next column. And then the extract, when there's no more blue component to collect, and we move it by one position to the right. So that's what we achieve at larger scale. So this is, instead of moving the column, and I'm going to go back to that slide. Instead of moving the, are we done with the video? Are we still seeing the video on the screen?
Nope, we should be all good.
Okay, so to achieve that movement of the column backwards, we actually move the input forward, which we simulate the movement of the column that way, hence the name simulating moving bed, if you were wondering. So why is SMB better than batch? First, you can see that we can process a lot more material. We don't need a complete separation between the two peaks. We don't need a full resolution. We just need a little bit of the front, a little bit of the tail being pure material, so we can collect that into the extract and raffinate stream. So that means we can fill up the column a lot more. So typically we get much larger load compared to batch. One, because we don't need to have a baseline resolution, and two, because we can use a lot more efficiently the packing material. And then we don't have an injection in the dead time to collect the product. It's a continuous flow in the process. So there's really an efficiency built in the process. The downside, it's isocratic only. The feed and the eluent are the same composition. You cannot have a feed in DCM and your elution in heptane ethanol, because eventually the DCM will be recycled in the system, and you will change your mobile phase polarity, and that changes your separation. Separation has to be perfectly stable all the time. So you have to have the same mobile phase, which means solubility becomes really critical because you have to use the solubility that your mobile phase is going to give you. And then it's a binary separation only. You get two streams coming out. So if you have more than two components, either you develop the process to have the component of interest first or last. So you recover either in the raffinate or the extract, or you have to do two steps. So the first step, you remove the front, for example, and then the second step, you remove the back end. There's alternate technique that are not truly continuous. The twin columns, the MCSGP and so forth, where you do use two columns to be able to achieve a cyclic process that provides you with the capabilities to process three peaks and get the peak in the middle, basically. But it's a very interesting process in that regard. So now that we understand the process, we're going to look at how we can develop chiral separation, because this is the main application, obviously, chiral separation being a binary system. And we're going to look at this. So the first thing is really looking at the feed material. Obviously, we want to develop this process on the feed material that's characteristic of the feed that we're going to process at the plant. So if we have a feed that's been crystallized 10 times over and has no impurity, and then we go to the plant and we see things that are a lot different, that's going to create some issues. So we want to be able to use something that's representative. Having said that, we evaluate solubility first. We look at different aspects of the product. Is there any insoluble in it? So with DSC, TGA, we look at residual salts. Is there water in it? Carl Fisher, etc. And then we look at the chemical structure. Do we need to add a modifier? Is it an amine that we need to add a base in a mobile phase, etc. And then is there reactivity potentially with different solvent? Like if it's a methyl ester, you may not want to use an ethanol-based mobile phase because you could potentially create an ethyl ester in the recovery process, which is distillation. So then once we have kind of characterized the feed, we can select the mobile phase for the screening. Now, why? Because now, since we have immobilized chiral stationary phases, immobilized means that they are permanently bonded on the silica backbone. We can use a larger variety of solvents, which means the spectrum of possibilities is becoming huge. So we need to narrow down the solvents that we can use. And then obviously we look at all the different phases that are available commercially, so that facilitate the scale-up. So solvent selection, obviously we want solvent that have a good solubility for the product, but you also want to look at something that doesn't have too much viscosity. So IPA is typically a good solvent, but the problem with IPA is very high viscosity, so that limits your flow rates. And then prep chromatography, batch or SMB, you always want to operate at the maximum operating pressure of your system. So if you have something that gives you high pressure already because of the viscosity, then your flow rates are going to be limited, which means your productivity is going to be limited. So things like acetonitrile, MTB are really good solvents for compounds, very good solvent in terms of hydrodynamic perspective, low pressure drop, also very good for evaporation. MTB, you have to watch out, it's low boiling point, so you have to watch out for emissions. But you also can use now with the immobilized phase, you can use ethyl acetate, DCM, 2-methyl THF, and so on. The list can be as long as you want. But all those solvents can be used on equipment because it's mostly stainless steel and Teflon PTFE-based material construction. So rule of thumb or tips for solvent selection, single solvent much better than anything because it's a lot easier to recycle. Ternary solvent is really if you have no choice. But if we have a binary mobile phase, 60-40 is preferable to a 98-2. Because 98.2, there's a good chance that a small variation on the mobile phase will change significantly the retention time. So the control of the solvent composition is going to be critical. It's going to be terrible if you have that variability in the mobile phase. Solvent recycling is very important. So we want to look at the properties of the solvent. The boiling point, is there an azeotrope? Can we recycle the solvent easily? Sometimes if the separation is at 60-40, but the azeotrope is at 70, we may actually want to run the separation at 70-30 because the recycling of solvent is going to be a lot easier. All right, so there's a lot of things to look into that. Then once we have identified separations and we do what good engineering work, we generate data that we need for modeling the process, modeling the separation. So we do loading studies. That allows us to get the data that we need for the liquid vapor, liquid solid equilibrium modeling. So loading studies are things of that nature, like you see on the right-hand side here. So you inject increasing quantities of product and you get the peak moving in either straight where you have linear isotherm here, or moving backwards when you have anti-Langmuir or S-shape isotherm. So that data can be used and model the process. And then we look at also the stability of the separation. So in this case here, we do every point is one analytical injection, and in between 2 points, there's been 10 overloaded injection to put a lot of material on the column. This is done on an analytical column size. And the idea is to monitor the retention time of the analytical to see if there's any evolution over time. Here in this case, we can see that the green is actually going down slightly. So that could be an indication that something is happening to the packing material. We actually having something that's strongly retained and changing over time the retention capability of the packing. And then we look at the transition from one step to the next. How can we evaporate to dryness? Do we have to do a crystallization step? What's the transition that we need? Once we have that, we have the model. With the model, we can do in silico optimization, get the idea of the productivity, and then we can compare with that productivity number, which is the number of kilo product we can process per day per kilogram stationary phase, which is the amount of packing that we put into the column. And that allows us to compare processes A to process B and see which one is the most performing for us. So 20 years ago, a productivity of 1 was a good productivity. Nowadays, we see productivity of 2, 3, and more. Some productivity north of 15 have been reported. The problem now is that you displace the problem into handling the volumes that you need to process, because now you're going to be processing a large amount of material in a small system. And all of that allows you to have a quality by design. You understand your process, you have a modeling of it, you have a good understanding of how your separation is going to go depending on how the parameters changes, the ratio of the enantiomers in the feed, the concentration, the mobile phase concentration, and so forth. So you can play with all that parameters in silico without having to use a lot of material. So some rule of thumbs for retention time when you develop a process. We don't want cycle time that are too long. So typically on a 4.6 by 250 or 1 mil a minute, we're looking at retention time that are within 10, 12 minutes for the second peak. The longer the retention time, the longer the cycle time on the SMB, so longer the switch time when we move the columns by one position. So that means the reaching steady state is going to take a lot more time. If the retention time is too long, we also going to use a very high flow rate in zone 1, which is limit the system pressure drop. And then if the retention time are also very, very short, maybe batch chromatography could be a good option for the process instead of using a lot more complex SMB system. Now, once we have all this, we can go on a small system. Here you have a picture of the mini SMB. We have 3 of those units on site here. They can use 4.6, 10 millimeter, and 21 millimeter columns. Also, that allows us to process 10 to 100 grams a day, depending on the size of the column and productivity. That allows us to separate material, optimize the productivity, look at the impurity profile, where the impurities are going into the extract of the raffinate, they're going to be recycled, where they're going to go. Is there any degradation over time of the separation, degradation of the product during recovery, all of those things that you need to know before you go to large scale. And then once we have completed this work, we have a really good idea of what's going to happen at larger scale. And then when you get to larger scale, obviously you go into manufacturing mode where you want to flatline a boring production where every batch to batch is reproducible, and you can do that for as long as you have feed and process the material efficiently, and becomes a routine. So how the system is set up at the plant, here you can see the 6 by 800 for the data on the side here. You can see this 165 kilo stationary phase. So if your productivity of 1, you can process 165 kilo feed per day in this system. So very significant amount of material. You have the SMB in the middle here. Falling film evaporators associated with the unit. Those are all continuous process. The solvent from the falling film goes back to the air tank for both sides. The air tank composition is adjusted with a makeup tank here. The composition is adjusted online by PAT, either capacitance probe or FTIR. and then the feed is made up in batch going into the process to feed continuously the SMB. The product goes into a storage tank until it gets to the dryers here, the conical dryers that I use a giant rotovap. We bring a solution in and we get a solid out here, extract and here raffinate. And then the solvent from those dryers go back into the makeup tank because this is going to be the other side of the azeotrope. With this process, you can actually recycle over 99.9% of the solvent. I have data I can show you in the next couple of slides. We typically have 95% on stream factor. That means the process is running non-stop all year long, and you can see the amount of material we can process in this kind of unit. Some pictures of the larger unit that we have on site is a 5 by 1000. The blue piece here is the jack that compresses the column. The column is actually on the bottom here. From the small scale to the large scale, all those columns are about 10 centimeter, 9 to 10 centimeter long. As I said earlier, we only scale by increasing the diameter, not changing the column length. We change the volume by the diameter, not by the length. So as a kind of a wrap up on the development piece, so the screening and the loading study takes about 2-3 weeks with about 2 to 5 grams of material, depending on the difficulty of the process. But very quickly, we can have an idea with the in silico optimization, what will be the best conditions for the separation, and what will be an estimation of the cost at commercial scale, depending on 10 metric ton, 20 metric ton, or 1 metric ton product. Then we can go into the proof of concept, and within a couple of weeks, we can confirm the productivity numbers, confirm the separation performance, and then generate material for evaluation and so forth. And then we can keep going, running the SMB, the mini SMBs as long as we want to generate more material to support development of the downstream chemistry and so forth. So you can see that very, very rapidly we can have an idea of the separation, if it's going to work or not, and if it's going to be productive or not. So obviously, nothing will work if we cannot take it all the way to the plant, to commercial scale. So we can demonstrate at small scale and gram scale here on the several units that we have on site. And then from here on, it's all GMP manufacturing. So we have a kilo scale unit, which is about 1 kilo stationary phase. We have the pilot, and then the 2 commercial scale units where we can do the transition. And we've done transition from those units many times over, with very successful scale-up performance. So now we know how to develop a separation, but what do we do with this? Where can we do the separation more efficiently? What can we separate and so forth? And then can we compare efficiently with other technologies? And can we combine racemization with the process? So let's look at a case calculation. Hypothetical case. We have here three steps of chemistry where we have a racemate that carry forward, and we do the chiral separation at the end. Obviously, this maybe we can do the separation at the step before. Why? Because now we don't have to carry as much material then to the next step. So it will save some raw material and obviously some cost. Obviously, if we can do it even earlier, what does that do to our process? So we look at pricing. Obviously, the first doing the separation at the end will be the most expensive because just the sheer amount of raw material and the volumes we have to handle. The second step will be less expensive, and the cheapest will be there, assuming that we don't have racemization in the downstream chemistry. Obviously, we don't lose the chirality. So obviously, we want to separate as early as possible in the process. Now, if we go a step further and we can find out that the C minus C that we're separating here, can we actually racemize it? If we can racemize it, now what happens is that the price point for this process drops significantly because now we're eliminating a waste stream in our process, and we potentially double the usage of the raw material that we have at the front end here. So the cost of that route should be cheaper than the cost of that route. And they all obviously going to depend on the productivity that can be achieved on the different routes. So now if we do some calculations, there's a bunch of hypotheses behind all this. But if you look at the step with our racemization here, the separation at step 2 is significantly cheaper than separation at step 4. It's just because of the sheer amount of raw material and volume that you have to handle creates more time and more volume at the plant. But when you do racemization, you can see that actually doing the separation at step 2 or step 4 is about the same price. So you will prefer to do the separation where you can actually have racemization of your process. And all of that allows you to also minimize your waste and your carbon footprint. So that's something to look into when you develop the process. Think about that separation early on in the process. Again, the same line of thought here. A lot of chemists love to use protecting groups. Some chemists prefer some groups than others. So maybe sometimes you can look at a different, especially in this case, we have a chiral. Maybe the protecting group can be changed to improve the solubility, to improve the separation factor, the productivity of the process, right? So maybe in the route, look at different options for your synthesis and look at that separation. And like I said earlier, the separation development doesn't take that much time. So once you have several compounds that you want to look at, it won't take that much more time to identify the best separation. Now, we always compare to tartaric resolution or asymmetric synthesis. So here we've done a tartaric resolution for many years before switching to SMB. So those are based on real data. So obviously tartaric resolution, you start with your racemic freebase, you make a salt, you separate the undesired from the desired, then you freebase your product and you get your chiral API. Here in this case, based on the process I'm looking at, we have a PMI at about 8 kilo per kilo of chiral pure intermediate. Now, what we do on the SMB piece, now you don't have to do a salt. You take the feed as is in the SMB, separate it. The solvent get reused, and then we've demonstrated that we can go almost 100% recycling. We get your product out here, so your solvent get reused here, and then the S isomer can actually be racemized because it comes out of the dryer as a solid, gets put back into a manufacturing plant, get racemized fairly simply, and put back in the SMB. So obviously the racemization. If it's a simple proton that needs to be pulled with a base, that's very easy to do. If it's a rotamer that you can just heat up to rotate, that's easy to do also. If you have to do a little bit more chemistry, you need to assess if that chemistry piece is cheaper than starting with fresh material. But you can see that the SMB process here, we basically have zero waste because we recycle the solvent and we recycle the design enantiomer. And then we look at the PMI, we have a PMI less than 1 here. And some data from that. If I look at year 2019, one of the many years we processed this material, we've done over 20 years, we've done over 1,800 batches of the API. In 1 year, we processed about 40,000 cubic meter, so 40 million liter of solvent in the SMB, but we only added 14,000 liter of fresh solvent in the process, which correspond to a recovery and reuse of 99.96% or greater than 99.96%. So basically that 14 cubic meter is only 0.04% of that 40,000 cubic meter here. So significant saving in terms of solvent consumption, carbon footprint, number of trucks on the road, waste generated, et cetera, et cetera. You can see the benefit of this recycling technology. Obviously, we also produce 85 metric ton of material in those 83 batches. It's about 1,000 kilo per batch. So when you look at the PMI, here's in terms of liter of solvent per kilo, because it's the main consumption raw material, we are way below a number of 1, which is extremely good in terms of any chemical processing. So you can see that this technology is sustainable, is very green in that regard. Quick word on racemization. I kind of talked about the atropisomer. We've seen in the past few years quite a lot of those compounds with our rotational isomers. We actually demonstrated on the separation on the pilot at over 50 kilo scales, and then we took some of the design enantiomer, racemized it through a 2-zone PFR system under CGMP. So you pump, heat up the material to the temperature where it rotates within a decent amount of time. Then you get a racemic material, pull it down before you go through your pressure relief here, and then you have your recipe material you can reprocess in the SMB. So those things can be associated in parallel with the SMB and make your process extremely efficient and cost effective. Now, this is the end. I'm actually a little shy of 45 minutes. So hopefully through this webinar, you've seen that chromatography is a flow process, a continuous process. So it has all the benefit of continuous process. It's smaller footprint for a large amount of material produced. It's fully automated, obviously less labor intensive. There's significant savings in that regard. It has very high yield, good potential for racemization, provides very low waste this way, simplifies the synthetic route. You don't have to go look at metal catalysts or complicated synthesis to achieve the purity. Racemic materials sometimes much simpler to make. The CSP, the chiral stationary phase that goes into the column, can be used for many years. We have done over 10 years for the larger system. If you know how to take care of it, if you handle your feed property and your material property, the CSP can be used for a long time. And then it provides you with a unique IP advantage because there's not a lot of people that can do this kind of technology. And then obviously it's, in my view, a very green technology. Even if it's chromatography, it's very, very green because the inherent capability to recycle, recover, reuse the solvent, reuse the undesired enantiomer, and so forth. So there's really a lot of benefit in this technology. Having said that, thank you very much for your attention, and we are ready for Q&A session. So thank you very much.
Ready for Q&A. Olivier, awesome. This was amazing stuff. I really appreciate the presentation, and I thought the sustainability stuff was great to see. I mean, the recovery is really impressive. We've got a bunch of questions in, so we're going to get to those in just a sec. Audience members, left side of your screen below where you're seeing me, there's a Q&A box. Submit your questions through that, and we'll get some answers here that we can get to. If we don't get to them all, I'll pass those along to the team again for potential follow-up after the event. On the right side of your screen, just two quick reminders. Olivier's information is there, so you can actually reach out to him directly just by clicking on the little blue circle on the right side, instead of having to write his email address or anything like that in there. And then below that, we've got three kind of related topics for today, three downloads that you can access and get some information on if you need anything else. So Olivier, I'm going to start with the Q&A. Let's go with this first one from Rob. So what is the demonstrated life of the stationary phase?
The
demonstrated life of the stationary phase.
So, like I said in the last few minutes, the stationary phase, we have had stationary phase for 10 years, being used 24/7 at commercial scale. I mean, over 160 kilo, 200 kilo packing in columns in systems. We typically have to repack a column once or twice a year because you have the inherent plugging all the frits with fine and stuff like that. But then the packing can be taken out of the column, decanted, and put it back into the system very, very easily, between quotes, because when you handle a large volume of stationary phase, you have to be a little bit careful. But we've done that demonstrated for 10 years. And after 10 years, we replaced the whole packing material because we found that the decantation was taking more time than actually replacing putting fresh CSP in the system. So if the key is to have a feed material that's clean, so you don't have impurities or things that get stuck on the packing that eventually degrade the packing. Now with the immobilized stationary phase, we've seen a lot of robustness. The solvents don't damage the packing as much as they used to be with the immobilized, the coated phases. So it is really a question of how much bad stuff you put on your packing will guarantee that you if you don't put a lot of bad stuff, your packing will last for a lot longer.
All right. Yeah, perfect. Thanks. Let's see another one from Tao here. So is SMB applicable to chiral separations?
Oh, yeah, SMB is typically the 99% of application of SMB right now in pharma is chiral separations. So separation of enantiomers. But we've done other things. We've done natural product. We've done impurity removals. We've done a separation. I remember a few years back where the crystallization was bad because we were losing a lot of product and we had to crystallize 3 or 4 times the material to remove an impurity that was closely related. We did it by SMB, and it worked very, very well. We had a good throughput, and it works well. So, but the main application is chiral separation, definitively. Yeah, that's what we talked about today. Yeah.
All right. Good stuff. Yeah. So I've got 2 questions here from Srinath. First one's from me. Will there be a link to the presentation shared for review? Absolutely. You can use the same link that you used today. Usually available after a couple hours, 24 at the most, but you should be able to get to it even if you wanted to watch this again later this afternoon or in the evening. You'll be good. Next one, also from, like I mentioned, from Srinath. Can SK carry out continuous chromatography of highly potent compounds?
So we do have the capabilities. We've had an SMB set up for potent compounds. That project does not exist anymore, so that SMB unit is not in use at the moment. But we have that capabilities, yes. Typically, those are smaller volumes, so the equipment is not a large volume, but we can do either batch or SMB actually for highly potent compound on our site here, and also on the batch side we can do in Ireland also.
Perfect. This one, so they're asking for your 20+ year customer process. Are you able to reuse the solvent for the next processing run?
So that's a good question. I would love to, but no. Typically, this is a very conservative industry, right? So the solvent is dedicated to a compound. Same thing for the chiral stationary phase. The chiral stationary phase is dedicated to a compound. Even if it's 2 compounds for the same customers, there would be 2 different lots of chiral stationary phase. No, the solvent is not reused for somebody else, unfortunately. Even if you can prove that it's pure past the raw material specification, there's still a lot of stigma in that area, and it's something we need to address in the next few years in this industry overall, if we want to improve the carbon footprint and the reusability of solvents and waste and minimize the wastes.
Yeah, no, perfect. That makes a lot of sense. Let's see. We talked about chiral separations. Can SM be used for non-chiral separations?
Yeah, no, absolutely. As long as you have one peak separates from the other impurities. Like I said, we've done a natural product where we had, depending on the season of the harvest of the plant that the compound that we were separating was an extract from the plant. You could have like 17 or 25 impurities the method was developed, so the impurities were actually bundled in the back end of the peak, and we were just collecting the front, which was the peak of product with a high purity north of 96%, and by simple crystallization, we're actually getting 100% assay on that material. So you can absolutely do it for non-chiral separation, for complex separations. It's just a question to tune the separation to where you want to have the impurity of concern or the troublesome impurity, like we like to call them, away from your main product, that you can collect them in a different stream from your product stream.
All right, perfect. So this is from Robert. Compared to like a traditional chromatography process, what's the cost of goods per gram look like for an SMB process?
So the advantage of the SMB is you can recycle significantly the amount of solvent. Typically, batch process, you will do a gradient with a water-based mobile phase, so it's very difficult to recycle that mobile phase. So typically, it's a very wasteful process in that regard. So the cost of goods I don't have the number in the top of my head, but I'm pretty convinced that the cost of goods by SMB is significantly lower than by traditional batch. There's no doubt in my mind.
Yeah, that perfect, makes sense. So let's see another one here. So what is preferred product in the raffinate or in the extract?
That's a good question I get all the time. So in batch, typically you want the product to be the first one coming out because you don't have the tail of the impurities getting into your product. But in SMB, actually the other way around, we like to have the product in the extract because it builds robustness in the process. But because in the extract, we're dealing with the tail of the raffinate going in there. So if there's a small shift in retention, the tail is not a big change in concentration. So your purity is going to change, but not significantly. Well, when you look at the raffinate side, it's the front of the extract that can come and collapse into the raffinate stream. Because it's a front, what's going to happen is the concentration is going to jump very quickly, and now your purity is going to go from 99 plus down to 80, 85 very quickly. So you need to be able to capture that with your in-process testing. So in that regard, we like the extract being the preferred product, which is the most retained. But sometimes we're no choice.
Yeah, no, makes sense. Another one here, I think it's Zishao. So how is the cost compared with the crystallization technology?
So I had in the table, it's an order of magnitude. So okay. It's a lot. It really depends on the productivity and how much recycling you can perform on the SMB. But on the tartaric resolution, we were easily an order of magnitude cheaper done by the batch tartaric process. Yeah.
All right, great stuff. Let's see. I think we can do one more here. So this one's from Akeem. Are you using chiral stationary phases, and how many are available on scale or regularly used on kilogram scale? And they're asking how expensive are these also?
So I will not comment on the price. They are expensive. Chiral's phase are expensive, but like I said, we can use them for a long period of time. So if you have for small scale, one time only, it's a high price. If you only separate a kilo or 2 kilos, right? So it's tough to absorb that cost. But when you run commercially hundreds of metric ton over several years, and you can use the same chiral stationary phase for that amount of material, when you do the calculation of dollar per kilo of product, you're down to single digit numbers, like a few dollars a kilo is the cost of the stationary phase. All the phases that we use for the screening that are in the screening slide, all of those phases are available at commercial scale. So if I need 20 kilo or 150 kilo or 200 kilo, I can get that from my suppliers, so either Regis or Chiral Technologies. Chiral Technologies has all those phases demobilized all the way to the IM series. All are 20 micron available at commercial scale in kilo quantities in bulk. So we will not develop something on a packing material that we have no confidence that we can get larger quantities of it. I mean, our big SMB system, the 5x1000, use about 225 kilo stationary phase. So the goal is eventually to be able to find that much packing material, right? So,
yeah, that's perfect. Maybe just one more here. So what should be the target chiral purity?
That's a great question. It depends if you can get chiral enhancement with the crystallization step afterwards. The higher the chiral purity, the lowest your throughput is going to be, because you have to have robustness in your process. So if I can get a 96% chiral purity instead of a 99, I may be able to run faster with a lot more robustness in my process, knowing that in the downstream crystallization, I can get the 96 improved all the way to 99 plus, right? So there's a little bit of development that needs to be done once we identify the separation. We've done a few first kilos separation. We typically go back and look at, okay, with a chemist, what is the minimum chiral purity you can get that your crystallization will still work and enhance the chiral purity. So there's like a combination between the two, and there's a point where you need to find that balance between the chiral purity that you demand on your SMB and the chiral purity you can achieve on your downstream process with the crystallization. So it's for every process, we shouldn't be looking at the unit operation alone. We should look at holistically at the whole process. So all the different unit operations, how they interact with each other, and how one can benefit to the next one.
All right. Maybe we can fit in one more here real quick. So Shui is asking, is SMB a more efficient selection to replace SFC?
So it's interesting because SFC is ubiquitous in labs in development for small scale, for gram quantities, 100 grams. We see a lot of projects coming our ways that were done first in SFC, work well, great technology, love it. We don't use it here because we don't have it, but I've been involved with SFC for a long time on the side. We see a lot of projects coming our way because the the SFC is limited in the number of kilos you can separate. And then now when looking at hundreds of kilos, there's a conversion from SFC to SMB, which is usually possible fairly quickly because CO2, methanol, or CO2 or organic solvent can be replaced. CO2 can be replaced by heptane very successfully. The advantage on the liquid side that we can use other solvents, so that helps in solubility and whatnot. So we see a lot of transition between SFC and SMB when there is larger demand in kilo separation. So,
okay. Perfect. I think it's going to be a good place to stop right at the top of the hour. Dr. Olivier Dapremont, thanks so much for joining us. This was great. We got through a lot of questions, and this presentation was fascinating. Audience members, thank you so much for joining us as well. We really appreciate your time here today. If you have any more questions, please submit them through the Q&A. We've got a couple we couldn't get to. I'm going to pass those along again to the team. And any more you submit, I'll pass along as well. And you can reach out to our speaker on the right side of your screen. So with that, have a great rest of your day, and we'll see you next time. Bye, everybody.
Thank you. Bye, everybody. Merry Christmas. Happy New Year.